One of the unique pieces of our study is that we used a screening approach in the mouse where we use CRISPR to manipulate genes and to introduce mutations in genes so that we can understand the importance of those genetic changes. People have done such screens on tumor cells within an animal, but I think our screen is unique because we are able to evaluate genes within a normal tissue context, which is really hard to study in cell culture or in cells in a dish, because usually those cells are cancerous cells. So when you're looking at diseases of normal tissues like the liver, you really have to study those tissues within the animal. And this technology allows us to investigate genetic changes of hundreds of genes at once in the normal tissue. And that gives us an advantage when we're investigating liver disease or things like tissue regeneration in the normal tissue context. So this is a unique tool that we're now going to use for future studies to examine not only mutations from human livers, but any genes that we're interested in to understand how they might impact regeneration. For example, we're using this to identify future drug targets that might be important in liver wound healing or tissue regeneration. And I think it's going to be incredibly useful for us to assess hundreds of genes at once rather than single genes one at a time.